Hello there, and welcome to today's Biology 101 lecture. Today we are going to cover the basics of genetics and cell division. Today is mostly about molecular genetics, and so next week we'll get into the consequences for how the genotype of something codes for the phenotype. But now we're basically just looking at how these things operate at very, very small scales. It kind of bridges the gap between our previous section on small things as we move into our next block of the class on more theoretical things. So let's dive right in here with Chapter 9, an introduction to genetics. So DNA contains the information for the production of proteins, which carry out a wide variety of tasks in living things. And so basically you can think of your DNA like a set of instructions that is made up of only 4 letters, A's, T's, G's, and C's. And you can write instructions in words, right, with the letters you have in English. You can do the same thing with DNA. And what these instructions do is they are instructions for a protein. They're basically the building blocks of that specific protein. Keep in mind, there are also regulatory genes that are not proteins, but today we're primarily going to focus on production of proteins. So we already mentioned we have adenine, thymine, guanine, and cytosine. These are the alphabet of DNA, and they are laid out in a double helix fashion, like so. Keep in mind that A's always pair with T's, and G's always pair with C's in DNA. This is going to become very important later when we look into replicating this DNA. Regardless of which side it is on, A's and T's are always together, and G's and C's are always together. And if you recall from the session before this, we talked about the protein production path, or how this is all going to happen. And really, it involves several parts. And let's go ahead and draw this on the board again, just in case you didn't quite remember, where everything is going on inside a cell. And within that cell, we have a nucleus. And within that nucleus, we have our DNA. And the DNA, although you saw chromosomes all over your chapters, is actually kind of spread out. There's still the chromosomes, but they're all spread out and kind of looks like spaghetti in there. And what happens is the cell receives a signal for producing a protein, and it exposes a portion of that DNA that's in here. A portion of A's, T's, G's, and C's in whatever order we want, and they come in triplets. Like so. And what's going to happen is when it exposes this portion, something called messenger RNA is going to come up and make a copy of this that's very similar. It substitutes U's in there instead, but it makes that copy with mRNA, and it sends that down to your ribosomes. And what your ribosomes are going to do is they're going to read this string of DNA, and as they read it, they're going to add an amino acid to the developing protein. And they can do this because each of these 3 codes for an amino acid. So, as they're reading along, like reading a sentence in a book, they're going to say, all right, this means add this amino acid. This one means add this amino acid. And they add these together, until a protein, you get a very large protein. And these can be thousands and thousands of amino acids in a row. And if you recall, we talked about these like Legos, right? We can put a bunch of different parts in here to build amazing things, because it's not just going to stay like a row like this. The protein is actually going to fold up into a 3-dimensional shape, and it's that 3-dimensional shape of the protein that's going to dictate what that protein can do. And so we see this kind of same thing that I just drew here in this figure, where we've got DNA being exposed so that we can take mRNA out of the nucleus into the cytoplasm here, where we're going to have ribosomes that are reading that DNA and making proteins. Most cells in an organism contain a complete copy of the organism's genome, meaning a collection of all its genetic information. Well, how does that happen? Let's take you, for example, right? You start off as a sperm and an egg, right? You have 23 chromosomes from your mother and 23 chromosomes from your father. And so, you start off that way as just one single cell. And you started off from there with the sperm, which had 23, and the egg, which had 23. When these two meet up, we end up with our 46 chromosomes inside this egg. Well, how did we get to having the exact same DNA in all the cells in your body? If you've ever watched a show with like crime scene investigation or something like that, what you'll see is that they're like, oh, we found a piece of hair, so we have their DNA, or we found something on the side of a cup, so we've got their DNA, or we found some skin cells, so we've got DNA. We found blood, we found semen, we found all these different things. And they all contain the exact same DNA. And so it's because of this fact that they all contain the exact same DNA that you are able to do that. And it's all because your DNA starts in here in this cell. In a moment, we'll go back to getting that sperm and egg, but right now we're just talking about this single cell dividing. Because it starts off at one cell. But you, as a large multicellular organism, are going to be made of many, many, many cells. The way we're going to get there is a pretty simple process. This DNA is going to condense into something called chromosomes. And if we look at our DNA, remember it was A's, T's, G's, and C's, and we can put whatever order of these we want here in our example, like that. And A always pairs with T, T always pairs with A, G always pairs with C, and so we can make our duplicate copy on down the road here. So how are we going to make a copy of this? Because right now, we're inside the nucleus of one cell, and we're just going to draw the cell here, like this. But we need to go into this cell splitting into two with two equal copies of the DNA. Well, the nice thing is that it's actually a lot easier than you might think. All we really have to do is split this double helix in half. So now we've gone from that winding staircase, that's a double helix, to two separate strands. And then to double this, all we do is we add what we are missing. So over here we're missing a T, A, C, G, A, T, T, A. And over here we just add what we're missing again. A, T, G, C, T, T, A, T. If you look at these two columns now, we have two identical copies of the exact same thing. All we did was we split our double helix in half, and we added the parts we were missing to each half that we had. And so then it's pretty much a simple division of the cell. We move this part of the DNA over here, this part over here, pinch off two parts of the cell, and then eventually they totally divide into two separate cells. And that's really what what the chapter on cell division was all about today. So, each of your cells has a complete copy of your genome. The thing to keep in mind about this genome is that you started out with one cell, right? You started off with that original cell that we were talking about. And how is it that you get so many different cells, right? A skin cell is very different from a cell in your hair, or a cell in your liver, or a cell in your heart. All these things are very different. Well, the trick is that you have all your DNA, and then you turn on and off certain parts of that DNA. So you start off, the ultimate stem cell is a egg that has just been fertilized. Because that cell has the potential to eventually become every single cell in your body. But what we do is we start to differentiate that cell and turn off the portions of the DNA we don't need. So as you develop and grow, what's going to happen is that some of your cells will say, I am going to be, let's say, connective tissue. And a type of connective tissue would say it'd be bone. And so it says, I'm going to be a cell in bone. And so it's going to turn off everything else for nervous tissue, muscle tissue, epithelial tissue. All these things go away, and it turns on that specific stuff for the bone cell. And so even though all your cells have the exact same DNA, they're only expressing or using the portions of it that they really need. So, we already talked about before cells divide, we copy the genome, and we make two daughter cells. And so really, your cells have to divide. You are constantly replacing your cells. For instance, you can fill up, you have a few liters worth of skin cells that you're losing all the time. Just like you have dandruff, your cells are constantly being sloughed off your body. That means you're constantly repairing cells on your skin and sending them to the surface to protect you, and then they're sloughed off and you make new ones and so on. Along those lines, when you grow, you don't grow by making your cells larger. You grow by adding cells to your body. And so you actually have more cells as an adult than you do as a little child. And so all this points to the fact that your cells need to reproduce in your body all the time. And so really, the process is as simple as I described before. You take your DNA, make a copy, move the copies to each half, divide the cell, and boom, you got two identical cells. We ideally copied that DNA without making any errors. We'll talk about those errors when we talk about mutations. And so we get double the amount of DNA, half of it is in each cell. DNA is packaged in chromosomes. And you see these chromosomes drawn like X's a lot and things like that. But in reality, your DNA is like that spaghetti that I showed you a little bit ago, where it's got to be open and the ability for it to be used, for it to do things. The way you can think about what a chromosome actually is it's a compaction of DNA for the purposes of moving it around during cell division for the most part. And so if you've ever taken a rope and you've twisted it, remember DNA is twisted, so if we take a rope and we twist it, you can start to get it kind of spirals. But if you keep twisting that, shoelace, rope, whatever, you keep twisting it, you keep twisting it, you keep twisting it, eventually there are twists on top of twists. And there are twists on top of those twists. And by the end, instead of having some long string, you've got this nice compact ball where that string once was. And that's what a chromosome is. It's really just twisting up and packaging that DNA so it's not spread out all over the place. When we are talking about chromosomes, so here's that twisting example. Here is an unduplicated chromosome, so this would be 1, and then we duplicate them, and this is 2 copies next to each other. Chromosomes come in matched pairs, okay? And such homologous chromosomes have closely matched sets of genes, and you as a human being have 46 chromosomes, 22 matched pairs known as autosomes, and 2 sex chromosomes. And this is kind of what a karyotype looks like if we spread all this out. But how do you get 2 copies of those chromosomes? Well, we already alluded to it a little bit earlier. Because what's going to happen here is that you get 1 set from your mom and 1 set from your dad. Okay? What do we mean by that? You start off with 2 chromosomes. Right? And I drew them red and blue for a reason. Because one of these comes from your mom, and one of them comes from your dad. But you only get 1 from your parent. So that means that your mom and dad both started out each with 2 of their own chromosomes for any given location. And so what I mean by that is this could be chromosome 1, 2, 3, 4, all the way to 22, and then your sex chromosomes for 23. You get one copy from your mom, one copy from your dad, but which one of these two do you get? Well, the answer is you get a hybrid. Because when there are these two chromosomes inside your parents, what they do is they mix their parts together. So let's draw them red and blue again so we can see this. And this is called crossing over, where what we're going to do is we're going to take some of this chromosome and we're going to mix it with some of this one. And we're going to take some of this chromosome and we're going to mix it with some of that one. And so now we've got two potential chromosomes that we could pass on to you. And really, at that point, it's just a matter of randomly which one ends up in that sperm or egg that ultimately ends up being you. Because you've made two, but only one of them is making it any further. And actually, when we talk about this in sexual reproduction, you end up making four, but we're not going to really deal with the intricacies of that today. You can think of it like two. So you send one on that's a hybrid chromosome, that's a mixture of your parents, or you can really think of it like a mixture of your grandparents' chromosomes that is sent on to you. And so you end up with a hybrid chromosome from your mom and a hybrid chromosome from your dad. And you do this for each of your chromosomes down the line. And that is how you go from being haploid, sperm and egg, to diploid, meaning you have a full 46 pairs. So let's move on here. Oh, there we go. And we kind of already talked about some of this. Meiosis is the process by which a single diploid cell divides to produce 4 haploid cells. And this is what we were just kind of talking about. These are called your gametes. Female gametes are eggs and male gametes are sperm. And they're the reproductive cells of human beings and many other organisms. And so here's an example of our two processes of cell division. In the case of mitosis, we end up making identical copies of our original cells. And so, as you can see here, you start off with 2N, meaning you're diploid. You make copies of that DNA. You line up those copies, you pull them apart to each side of the cell, and you end up with 2 identical cells. That's the one we already talked about very early on. That's mitosis. In meiosis, we do something very similar to this, but instead, our ultimate goal is to work through this to make gametes. And so instead of ending up with 2 cells that are 2N, we end up with 4 cells that are 1N. 1N meaning each of these is diploid. So if this was a human cell, each of these would have 23 chromosomes. And here they're showing you the process that goes on. One of the significance of this is 2 things. There's diversity that comes about because these daughter cells are not exact copies. And really, 2 major ways this happens. The first one is because of crossing over, and that's just what I showed you up here a moment ago. Well, what we're going to do is we're going to take both your chromosomes before we make a gamete, and we're going to swap around genetic information between them. Keep in mind, the swap is pretty much equal. So both of them that you make end up with the appropriate genes. So it's not like one of them ends up with 2 genes for eye color and the other one has 0 genes for eye color. Both of these will end up with a gene for eye color because both the starting chromosomes had a gene for eye color, and you make a hybrid and it ends up with one or the other. So that's crossing over. And then the other thing is it's just random which one of the hybrids that you've made ends up in the sperm or the egg. And so this means that what we're doing is we're generating a lot of unique diversity for evolution to act on. Because if you think about this, no 2 people other than identical twins look exactly the same. And that's because when this crossing over occurs, you end up with a unique chromosome that has never existed in the history of the world. It is totally 100% unique. And then you have one unique chromosome for mom and one unique chromosome for dad, multiplied 23 times, put inside a sperm and an egg that are going to come together. And so you take all that uniqueness and then yet you combine it together to make even more uniqueness. And this is why in sexually reproducing organisms we see such a great diversity of traits. No two people look identical. They can look similar, but never identical. And it also goes back to our crime scene things that we mentioned before. Because you're so unique and identical, that's why we can identify you with that DNA we found at the crime scene. So, you have 23 matched pairs of chromosomes, as we mentioned before, and one pair of sex-determined chromosomes. In females, they're X's, and in males, you have an X and a Y. And really, you can look at it this way. Look at the difference between these two. The female X is very large and contains a bunch of stuff, and the male Y actually is much smaller, and it's missing some information that is on this X. You'll read in your book a little later about traits that are associated with the fact that there's this disproportionate amount of genetics between the two of these. And no matter what you've heard before, sex of an offspring is determined by which sperm gets there. A sperm with an X chromosome or a sperm with a Y chromosome. Any myth you've heard about eating something or doing things a certain way, whatever, all of that comes down, doesn't matter, because it all comes down to which sperm actually makes it there first. That is the only thing that will determine the sex of the offspring. And so we kind of spent some time talking about humans. Let's look at some other organisms for a second. Not all reproduction is sexual. Fusion of two reproductive cells. Most organisms are capable of asexual reproduction, primarily because most organisms are not large multicellular animals. And one form of asexual reproduction is binary fission. All binary fission is what we mentioned before, where if you're a bacteria, what you're going to do is you are going to replicate your DNA, move aside to each part of the cell, and split in half. And that's it. Bacteria reproduce that easily. And they made two identical copies of themselves with the exception of possible mutations. Other forms, bacteria can produce through, we already said binary fission with bacteria. Plants can use something known as vegetative reproduction. And what that is if you've ever seen somebody take a part of a plant and create a whole new plant from that, that's vegetative reproduction. And it's kind of a similar concept to regeneration, where you can take part of an organism and the rest of the organism results from that. Your book here talks about worms undergoing regeneration. Worms do not really undergo regeneration at any sort of meaningful rate. Most of the time, if you cut a worm in half, it's going to be dead. You don't end up with two new worms. Really, because that worm has a head and a tail. It's largely possible for a worm to regenerate the back end of its tail, but if you cut it through anything meaningful, it's dead. And so worms really do not rely on regeneration as a form of reproduction. It's more of a way that they can help survive after an accident. And here's an example of a sea star. This is actually a closer to true regeneration. This has broken off here from a sea star and regenerated a new little part, and you'll see here the extra parts that it has there. All right, the double helix. We've already mentioned the DNA is shaped like a double helix. This research was finalized by Watson and Crick, but as with anything in science, they built on the knowledge of previous scientists. And at the time, one of the things that really led to their breakthrough was research done using X-rays that had revealed the structure of DNA. It's a very complicated process. It's not like the regular X-rays that you would take to look at your bones in the hospital or something. And this was done by somebody named Rosalind Franklin. She was able to show her results to Watson and Crick before she even published them. And this kind of gave them the insight to do what they did. And it hasn't been until relatively recently that Rosa Franklin has been given credit for her contribution to discovering the double helix, although it could not have been done without her insights and research. So, the components of DNA and their arrangements, we've already hit on the basics of this. A with T's, G's with C's, double helix spiraled together, looping up like that. Exactly what you saw pictures of in your book many, many times. And DNA replication, we've already talked about this as well. The double helix divides down the middle, we add what we're missing, and we end up with two identical copies. And this is what I drew you closer to the start of the class. And we already mentioned how this leads to protein production, because each of those series of 3 base pairs codes for an amino acid, and we end up with what we needed. But let's take a moment to talk about mutations. A mutation is nothing more than an error in the replication of that DNA. And so you may have heard the term mutations used in popular culture. Maybe with superheroes and you heard like, oh, they're mutants, so now they can shoot lasers from their eyes and control the weather and stuff. That's not how mutations really work. Mutations really work in a very simple way. Let's say there was a string, we'll just do one strand of the DNA. There was A, T, T, G, C, C, A, T. All right, and so each of these is supposed to make an amino acid. And we put those amino acids together to make some protein. And let's say that this series went on and on and on, and it was supposed to make a protein that was like this, shaped like that. And let's say this is an enzyme that's job is to break up carbohydrates, like we've mentioned before. And so this is its very specific shape, and so this is exactly what it's supposed to do. It's okay at its job. And so one day, though, a cell is copying, and there's an error. Instead of making a T here, it adds a G. Well, now we've changed that amino acid. There's a couple things that can happen. One, perhaps this is what's known as a silent mutation. There are 64 possible sequences of A's, T's, G's, and C's, but there's only 20 amino acids, so there's some duplication. Sometimes a switch like this can have no effect at all, meaning it's a silent mutation. Another thing that can happen here is maybe we change it to a C, and it causes a change in the shape of our protein. And maybe now, instead of having a protein like this, we end up with a protein like that, and that enzyme is shaped wrong. And so now, whatever organism has this mutation produces enzymes for breaking up carbohydrates that are misshapen and can't function as well. What do you think is going to happen to the survival and reproduction of an organism that has this misshapen enzyme? Well, if you think about it, this one can have a great life. This guy can go, we can find carbohydrates, digest them easily, get lots of energy, doing well. This one, this probably doesn't function very well, if at all. And when it doesn't function very well, if at all, this guy can't digest carbohydrates as well. They don't survive and reproduce as well. And this mutation dies out, never to be seen again. Another possibility is that we totally lose a base pair. If that happens, that shifts the whole reading frame. So everything, every single amino acid after that point on is off. It's wrong. And so instead of producing something that's shaped like this, in that case we're going to produce something that looks like that. A bunch of nonsense. Doesn't function at all. that one dies out too. Most mutations will be neutral or deleterious such that they reduce the survival and reproduction of that organism. But sometimes, in very rare events, we end up making a change that does something good. And maybe, if we're taking our enzyme here, maybe this change in the DNA makes this enzyme, instead of looking like that, look like this, and maybe it adds, or let's actually do this differently. Maybe it just bends it ever so slightly like that. Between this one and this one, all we've done is made a very small change in the shape of our protein. But what if this very small change in shape actually makes this enzyme function better than this one? How do you think that's going to influence the survival and reproduction of an organism with this enzyme? Well, if you think it's going to increase it, you're right. Because if this organism is better at digesting carbohydrates, let's say it gets more energy more easily from carbohydrates, it will have better survival and reproduction, and this enzyme will become more and more popular, or more copies of it will show up in the population because of the better survival and reproduction of organisms with that enzyme. If that's not totally clear to you, don't worry about it quite yet, because we'll be talking about this when we talk about genetics next week and ultimately evolution, how these mutations can lead to different traits in organisms and ultimately new species. But for now, it's enough to know, change the code, change the shape. Most of the time, almost always it's bad. Very rarely, it's something good that will proliferate in the population. Oops, got to slide over here again. Okay. Where do we leave? There is our mouse. And so this is exactly what we were talking about. We're just losing or connecting the wrong base pair there and a point mutation. And most mutations have no effect, or if they have an effect, it's negative. And cancers are a form of mutation. Cancers are basically, and remember how I told you earlier there were some regulatory genes there as well? There are some regulatory functions that tell a cell when it should divide and reproduce and when it shouldn't. Cancers result from mutations that cause those regulatory genes to be locked in the on position. So instead of having a switch where you can say, okay, let's replicate now, let's not replicate, let's replicate now, let's not replicate, moving that switch back and forth, instead you end up with it locked on. And when that gets into the locked on position, what's going to happen is that cell's just going to keep reproducing. It's going to start with one cancerous cell, goes to 2, goes to 3, 4, 5, and so, sorry, goes to 2, goes to 4, goes to 8, 16, 32, and so on. Those cells keep replicating very, very rapidly. Another problem is that your immune system doesn't always do a good job of picking up on that. Because it's your immune system's job to find foreign invaders, as well as sometimes your own bad cells. But in this case, they're your cells which have your genetic markers, and so your body is not very great at fighting off cancer most of the time. Because it recognizes those cells as part of you, and so it does not want to attack them. Cancers have one mutation to start growing wildly, but then if they want to spread, they have to have additional mutations that allow them to end up in additional tissues. And so, for instance, if you had cancer in your liver, it's got to spread through your blood or maybe your lymphatic system, and being able to survive outside that liver area requires a mutation. And then you have cancer cells spreading around your body, and eventually there's a mutation that allows them to implant and live in other tissues. Until after a while, you basically have your own cancer with about 50 different mutations that could have invaded your body. Your cancer is unique to you. It is your cells causing problems that ultimately lead to cancer. Now, in this case, because they're your cells, cancer is not a communicable disease. You might be scared of hugging somebody who had Ebola, but if somebody says they have cancer, you're not going to be scared of hugging them. And that's because we can't transfer cancer from person to person. There's only one instance, which is kind of a big exception to this, and this was in, well, there's several instances where crazy things have happened, but this is one of them. This was in Tasmanian devils, and Tasmanian devils fight a lot. Like, if you've ever seen the cartoon, that's really what it's like. They will tear each other's faces up. And what they were finding is Tasmanian devils with tumors all over their faces. And they couldn't figure out why this was. They looked for viruses. They looked for bacteria. They looked for a bunch of different things. And what it turned out was that they were transferring cancer from the face of one Tasmanian devil to the face of another Tasmanian devil. But this shouldn't have been able to happen. Because if tissue from one organism gets in another, their immune system should fight it off. But in this case, what was weird is that the Tasmanian devils had gone through a population bottleneck, or a population crash, where they had gone down to very few individuals in that population. And what that meant was that all those individuals were very genetically similar. And because they were genetically similar, when they got in fights, they would tear open each other's faces, and if one had tumors, some of the tissue from their tumor would get in the other one's face. And when that happened, those rapidly growing cells would start to rapidly grow in the other Tasmanian devil's face, but the immune system would not fight them off well because those cells were so genetically identical to their own that the immune system did not really recognize it as that foreign. And so that's just a little story to kind of link together some of these concepts that we've been talking about today. Another key thing to remember about mutations and cancers is that the only ones that are heritable are those that occur in your sperm or eggs. Okay? If you get skin cancer, that's a mutation of these cells. But it's not that cancerous skin cell that goes on to make the baby. It's the cells generated by your gonads and ultimately your gametes that come together to make that new organism. And so only mutations in your germ cells can be passed on to the next generation. Now keep in mind, once that mutation occurs, once that sperm and egg get together, that individual has a mutation. That individual has that mutation in all their cells because it was in that first cell that started their sperm and eggs. And remember, you come from one cell. And so because that started with those sperm and eggs, it was in that first cell, it's in all their cells after that, including their gametes, so they can pass it on. Another thing that we hear about is, when we talk about mutations, is mutagens. You can also think of these oftentimes as things that cause cancer. Cancer is not the only mutation, but it's the one we really notice. Substances such as cigarette smoke, ultraviolet light can mutate DNA. And normally what's supposed to happen in your body is that you have, when you're copying your DNA, you have specialized enzymes that are supposed to go back and double check to make sure you copied it correctly. And if you don't copy it correctly, they're supposed to fix it. Sometimes those fail. Some people have a predisposition to cancer because those regulatory genes that check for errors don't function as well as those regulatory genes that check for errors in other people. And so if your regulatory genes don't function as well, you're more likely to get cancer. And this brings us up to an important point about mutations and cancer. And that is that a given mutation, cancer is not a guarantee if you're in contact with something that's cancer-causing, a mutagen. For instance, think of cigarette smoke. Of those of you watching this, how many of you know somebody who has smoked and got lung cancer? Possibly. How many of you know somebody who smoked a pack or two a day and never got lung cancer? How many of you know somebody who never smoked at all and got lung cancer? All of these scenarios are possible because all that mutagens do is they increase the likelihood of you developing cancer. What I mean by that is if you smoke, you are more likely to have an error in DNA replication that is cancerous and causes cancer. It doesn't mean it's a guarantee. There are very few mutagens which would guarantee cancer. I mean, unless you went and wanted to hug some spent fuel rods from a nuclear reactor, you'd be dead pretty quick with that. probably within a day, if you got that close. But most mutagens that we encounter in our daily lives increase our chances, sometimes a little bit, sometimes a lot. And we already mentioned this, but when we talked about these mutations, but mutations are going to be important to evolution because they provide new genetic information and new phenotypes on which natural selection can act. And so we create new things, new novel things that maybe haven't existed before through mutations, through changing that code. Most of the time those will be bad and die out or neutral, but sometimes they're good and they get passed on. And we'll talk more about this when we talk about our chapter on evolution. And so let's get to the last portion of our presentation here. The structure of proteins, we've kind of already covered it, so we're not going to really belabor it here. We've got amino acids. We take those amino acids, we make a polypeptide chain by stringing them together, and we ultimately fold that into a three-dimensional shape. Some proteins that we make are brought about through taking two separate chains and putting them together, and that's how we get our proteins. So that has kind of been our trip through molecular genetics. Next week, we'll talk more specifically about how genes reside on those chromosomes and how your genotype, or the specific genes that you have, ultimately